Before I move on to the rest of the topics, there is a small debt that I have from the previous lesson. One of you asked me whether U-test necessarily reflects the median of the population, and I thought, like, intuitively I thought it wasn't, but I didn't have a concrete answer, so I thought about it a little bit, and the answer is no. So, at least in theory, you could have the U-test goes to one direction and the median goes to a different direction, and I give this simple example here. So let's say we have two populations. One with values 1, 2, 3, 50, 101, 102, and 103. And the second one with 11, 12, 13, 49, 111, 112, and 113. So in this case, I hope you can see that it's quite obvious that the U-test will give a higher median to, will say that the second group is larger, because you can clearly see that most of the values on the second group are larger than most of the values on the first group. Where the one exception is the value in the middle, which is the median of the two groups. So in the first group, we have a median of 50, and in the second one, we have a median of 49. So actually, the median in the first group will be larger. Okay, so this is a very, of course, very artificial and very pathologic example, but I think it illustrates that at least theoretically, you could have U-test not necessarily reflecting the median. So like I said, it checks something somewhat different. It doesn't look at the actual values. It just looks at the ranking of the values. And it somehow aggregates all the values together, unlike the median, which just gives one value to each group. So these are somewhat different things. But of course, in real life, in most cases, U-test and t-test and the median will typically tend to go together. OK? So let's continue from where we stopped before the break. So we presented ANOVA, and I want to spend a few minutes speaking more about the assumptions of ANOVA. So like with t-test, we said that we assume normal distribution of the means in all the groups. But like with t-test, we tolerate some deviations. Like with t-test, we assume equal variance in all groups, and again, we tolerate some differences in variance, which are not too big. And there is even a more sophisticated version of ANOVA which spurs this assumption. We of course, like any test, assume independence of observations, which is crucial within groups. And like with t-test and u-test and all the rest, we must have that the groups are mutually exclusive. And if that's not the case, we must make it so by defining the groups in a proper way. OK, and there are some extensions of ANOVA that some of them mitigate these assumptions. By the way, Chen, I think it was you asking me about which things are not implemented in Python. And here you have one example. So unfortunately, the Welsh version that deals with unequal variance is not implemented in Python. But again, like I said, I think most of the time it's fairly robust. And you can take other implementations if you want. And there is also the two-way ANOVA, which is another extension that can examine the influence of two categorical variables on one continuous variable. So it's a more complex version. But we won't go into it in more details. And let's do a demonstration. OK. So we want to compare two populations. As an example, we will take the example that we mentioned of comparing the genome length of DNA and RNA viruses as we extract from virusome. So I wrote some code here that extract the relevant viruses and creates this box plot, which you can see for the 217 RNA viruses. And for 167 DNA viruses, you can see the distribution of the genome length, and you can see it in log 10. Because again, this spans many orders of magnitude. And if we want to look at it within the same graph, then we probably need to use a log scale. I won't go over this code example. It's things we covered many times in the past, so just pandas and matplotlib kind of analysis. But OK, so intuitively, we see some difference. We see that DNA viruses have a much wider distribution, and they seem to have a much higher mean, which you see as the blue bar, and a median, and the mean, which you can see as a blue rectangle, triangle in the middle. So in both measures, you have that DNA viruses appear to be much larger than RNA viruses. And we want to know whether it's statistically significant or is it just due to random fluctuations. So let's begin with a simple test of t-test and Mann-Whitney. So again, just import them from SciPy. And for each of them, I get the statistic and the p-value, which I actually care about. And you can see they give pretty much similar results. So the t-test is a little bit more significant, and the u-test, as typically is the case, is somewhat more conservative. But either way, we get a very significant p-value. So I think it's safe to conclude that what we see in the graph and what we see in the statistical analysis, whether we use a t-test or which is sensitive to distribution issues, or the u-test, which is much more robust to what exactly the distribution is, either way we get a very significant p-value. So we conclude that there is indeed difference, and DNA viruses are indeed larger than RNA viruses. Now, so here we saw a very clear example of very different means and medians between two groups. But what happens if we force the two groups to have the same mean and the same standard deviation? OK, so what we do here, we write this normalize function, which given any values, we'll just convert it into z values. So if you remember, z value is just subtracting the mean and dividing by the standard deviation to get the same values, and forcing them to have a mean of 0 and standard deviation of 1. So we do that for RNA viruses and for DNA viruses. And you can see that now the distributions look a lot more similar. Of course, we forced them to have that. So you can see that both have a mean of 0, because that's what we forced them to have. And overall, they seem quite comparable, but you do see some differences. I mean, they don't look exactly like the same distribution. For example, you can see that with DNA viruses, you have some outliers, which are larger. And with RNA viruses, you also have outliers, but less extreme than with DNA viruses. So it does seem like the DNA virus has a heavier tail. And then you can ask, OK, what happens if we try again the t-test and the u-test? So the t-test, of course, is not significant. It gives a p-value of 1, which is expected, because we forced both of them to have the same mean. And that's exactly what t-test tests. It tests whether they have the same mean. And of course, they have exactly the same mean. So we get a p-value of 1, which is obviously not significant. The u-test is a little bit more complicated. I mean, it could, in theory, provide us a significant p-value because it compares something different. It doesn't exactly compare the means. Rather, it compares which fraction of which group is larger or smaller than the other groups. But again, the u-test is not significant either. So we don't see that one group appears to be larger than the second after we normalize them, which is not very surprising. But we could still ask, OK, does it mean that the two distributions are perfectly the same, or might there be other kinds of differences? And if we want to test that, we can use the Kolmogorov-Smirnov test, which I import here. So now I can give it the normalized RNA genome lengths and the normalized DNA genome lengths, and compare the two of them using the Kolmogorov-Smirnov test, and get a p-value. and you see that the Kolmogorov-Smirnov test is significant. So even though we normalize them, they still have different distribution, and now we have a statistical analysis which proves it. Of course, the downside is, like I mentioned, with Kolmogorov-Smirnov, that it's not very helpful in an intuitive way. So we can say that it's not perfectly the same distribution, but how exactly do the distributions differ? It's quite hard to tell. But we only know that they aren't exactly the same.